Last time we have introduced the mechanism of virus infection and coronavirus outbreaks in Wuhan, China. Today we will talk about potential treatment. A recent reported cure case of coronavirus infection from Snohomish County, Washington, was treated with a new antiviral drug called Remdesivir. Is it actually effective for the novel coronavirus? There is a basic rule to judge whether a medication is effective for a disease. That is, we need to do control trials. For example, there is a patient who took a medication. We are not necessarily sure that this medication is effective because it is possible that she could be better off without the medication. So we should design a control trial to have two people with similar symptoms. One takes medication and the other doesn't. And then we compare their changes after a period of time. In practice, two people cannot be enough. We need a lot of people to do this kind of control trials. The first controlled clinical trial was in the UK in 1747. Dr. James Lind conducted one of the first ever controlled clinical trial on scurvy. At that time, scurvy was a disease that many crew members would get when going out to sea. Nobody knows why people get scurvy after a long time in the fleet. When James served with British Navy, he found 12 sailors who got scurvy. The 12 sailors were divided into 6 groups, with 2 sailors in each group. And then they were treated with different sour food to see which group will live better. Each group were given apple juice, diluted sulfuric acid, vinegar, seawater, oranges and lemons, garlic and mustard every day. After a period of treatment, James Lind found that only the group given oranges and lemons were fully recovered. So he concluded that the fresh fruit could treat scurvy through this control trial. Scurvy is a disease now known to be caused by vitamin C deficiency, but in Lind's day, the concept of vitamin was unknown. The control trial can be used in many fields. Let's take another example: the relationship between smoking and lung cancer. Is smoking related to lung cancer? In the early 20th century of Britain, only a few hundred people died from lung cancer. But after the World War II in 1947, nearly 10,000 people died each year from lung cancer. So British doctors wanted to know why so many people died of lung cancer in such a short period. In the beginning, people were skeptical of air pollution after the Industrial Revolution. But then they suspected more and more people smoking were probably one of the reasons. But how do we prove that smoking can lead to lung cancer? 2 English epidemiologists, Austin Hill and Richard Dole, conducted a controlled clinical trial. This trial lasted for many years. From 1948 to 1950, they have a hypothesis that the more people who smoke, the more likely they have lung cancer. He ranked the number of cigarettes people smoking, and it turned out that more people smoke, the greater the proportion of this population having lung cancer. It was finally written as a report with conclusion that smoking would increase the chance of getting lung cancer. However, just such a report cannot convince the public. Then he did a survey from 1951 to 1956. This survey is based on 60,000 doctors in the UK. He sent out questionnaires to many doctors asking if they smoke. And then he followed up on health condition of those doctors. This survey further strengthened his initial conclusion. The probability of lung cancer is much higher in people who smoke than those who don't. The results scared Richard Dole himself, so he quit smoking immediately. He published 2 papers in 1956, but this data collection didn't stop until 1993, when half of the surveyed doctors had died. According to the World Health Organization survey, there were 3.3 million people died from smoking every year. This is worse than almost any infectious disease in the world. So quitting smoking early is good for yourself and others. Now let's go back to the drug we started with, the antiviral drug Remdesivir. Can we use Remdesivir to treat patients infected with the novel coronavirus? Again, we need a clinical trial to study that. Generally, a clinical trial is divided into many phases. The first phase tests of safety. The goal here is to determine what the drug's most frequent side effects are. Once the first phase of safety is passed, we enter phase 2 and phase 3 to test for effectiveness. During this 2 phases of trial, we will use the control trial dividing patients into 2 groups for comparison. A stricter comparison methodology is introduced, blinded experiment. In medical science research, the concept blinding includes single-blind, double-blind, and triple-blind. Let's talk about the difference between single-blind and double-blind. Single-blind studies blind patients to their treatment allocation. It means that patients are randomly divided into 2 groups. One group receives the new drug and the other one receives a placebo. The placebo looks exactly like the drug, same color and same dose. Patients can't tell whether they are receiving the medication or the placebo. Why do we do that? Because the psychological cues are also important. On one hand, if the patients know that they are in the control group and not receiving any medication, their negative mental status might cause the disease to progress. On the other hand, if the patients know they are taking the medication, their positive mental status could help with the cure. In order to prevent these factors, a placebo will be given to the control group. This is called single-blind. A patient does not know if he's in a control group or a treated group. Double-blind studies blind not only patients, but also doctors who allocate treatment. In this case, the doctor don't know whether a patient belongs to a control group or a treated group, because the information a doctor gets will also affect his judgment and behavior. It also affects the fairness of the trial. After the entire complicated clinical trial process, a drug can finally be certified by FDA. It will probably take more than 10 years and hundreds of millions of dollars to develop a drug. Now let's talk about the company which developed Remdesivir, Gilead Sciences. Gilead first used Remdesivir as an anti-Ebola drug and it failed in the trial against Ebola. The research didn't stop there. Remdesivir is used to interfere with virus RNA synthesis. The cronavirus is also an RNA virus, as we have introduced in one of our previous videos. It has been found that this drug was effective against coronavirus, including SARS and MERS. Although Remdesivir is not FDA certified at this moment, there is a policy called compassionate drug use. It means that using an unapproved drug to treat a serious ill patient when no other treatment are available. There was a patient recently infected with the novel coronavirus in the United States. After taking this medication, the patient felt better the next day. This drug seemed to be very effective, but only one case cannot prove its effectiveness. So Gilead hurried issued a statement, saying that they did cure an American patient with Remdesivir, but this medicine has not yet licensed or approved anywhere globally. On February the 5th this year, the registration for clinical trial on the antiviral drug Remdesivir has been approved. It's a collaboration between Chinese health authorities and Gilead Sciences. A total of 761 patients infected by the novel coronavirus has been enrolled in a double-blind randomized control trial. This process probably will last until the end of this April. So the process to determine whether a drug can cure a disease is very complicated. It has to go through a long clinical trial, and we cannot conclude the effectiveness of a medicine due to one single case. Ridiculously, some people claim that because alcohol can kill virus, so that we can drink a lot to cure the disease. Do you think drinking alcohol can help to kill virus? Leave a comment and let us know.